Hello everyone, welcome to the ITLS Academy. Today we are going to discuss about the basics of quality control. So this quality control is not a single step. Okay, we prepared the entire product and then we are going to maintain the quality. We need to maintain the quality at every step, either the selection or acceptance of raw material or for the manufacturing we again required quality control. For product testing we required quality control, and the packaging we required, and after the packaging, when the product is completely ready for the marketing, again we require the quality control. So it is a huge process which is regulated at every step. So this is a basic definition of quality control. Again, it include not only the analytical testing of finished good products but also the assessment of operation, beginning with the receipt. Again, what I told you about earlier, receipt of raw material, the very first step for the manufacturing of anything. Then continue throughout the production, like obviously we select the raw material, we are going to produce something, and then we after production we go with the packaging operation, then finished product testing. Like after production of the final product, we again go with the testing, documentation. Yes, for the marketing we require documentation, and then surveillance and distillation. So this is an entire process in which every single step required are quality control. So we need to maintain it. Now, here's quality assurance. See, this is another term other than quality control. Recently we studied about quality. What is quality control? That it is a procedure which is required at every single step of the manufacturing, from acceptance of raw material to its manufacturing, production, documentation, surveillance, marketing, etc. Now we are going to know what is quality assurance. See, quality assurance clearly mentioned. Quality assurance play a central role in determining the safety and efficacy of medicine. Like efficacy after the final production of the product, how that medicine or any product which we prepared is highly efficient or not, is highly efficient for the marketing or not. We are going to know about this. So it is highly specific and sensitive analytical method. Again, important, analytical method. It's a kind of analytic. We need to analyze and then we reach to the final conclusion. Analytical method holds the key to design. Basically, obviously, if we are preparing any kind of product, first of all we require the design, and then development of that particular design. We need to maintain the standard. In quality, standard matters very much. Then quality control of the medicinal product, yes. And then total quality control will include all those aspects, starting with the procurement of raw material to the finished product. Obviously, I already told you about that, how the selection procedure go from the raw material up to the finished good products. The very first is we need to select the raw material, then we require design, we need to develop that, maintain the standardization. After manufacturing we go with the final product available at the drugstore until consumed by the patient. Obviously after the production these are going to the medicinal shops, and from these medicinal shops we can purchase that. So it reaches by the same procedure to the patient. Thus, it will include not only the parameter of good manufacturing practices but also storage, handling, and preserving the sample till ultimate use. Obviously, we know if we purchase anything, it is not that we can use it immediately. It can be kept for at least 1 month, 2 months, 3 months. Basically, their manufacturing date to their expiry date. That is clearly mentioned on any medicine, that what is their manufacturing date and what is their expiry date. So expiry dates are designed that up to what time we preserve that medicine and up to what time this medicine is efficient for the patient use. So that is, and also mention on that box that where we need to preserve. Somewhere mention on 4-degree refrigerator, in dark, etc. So we need to go with that procedure if we want the full efficiency of that particular medicine. So this is all about quality assurance. Remember, quality assurance and quality control, these both are two different terms. Now we are going to know what are the major areas of quality control, like what are the areas which quality control can cover. The very first is are good quality and nature, very very important. If we need to purchase something, we always prefer good qualities. Physically and chemically pure. Physically, what this means, physically and chemically pure? Physically pure means the selection of raw material which we select. We need to select raw material very physically, like the raw material which we select is contaminated, having other than our desired product, so that is a physical product. And chemically is when we start manufacturing in the lab, so that is chemical pure. So these are the two basic terms, physically and chemically. Physically, up to the product not reach to the lab, and chemically when the product reach to the lab, that is a kind of physically and chemically pure. And then we go with contain amount of ingredient as stated in the label. We clearly say on the any medicine cover, or that what are their ingredients, what ingredients are present, like phosphate, amount of phosphate, sulfate, amount of sulfate, and the sodium chloride if present, what is the amount of sodium chloride, barium chloride, etc. We always on the packaging it is clearly mentioned what ingredients are present and in what amount these ingredients are present. So we need to go with that, and it is to be effective after administration. Very very important that administration is what, when we go for the documentation of that particular medicine. If I prepared something, if I manufacture something, I know I need to go with the documentation. So only till that time our medicine not be efficient. It need to be efficient till the patient it consume. So we need to maintain its effectiveness, and then it retain quality in terms of shelf life or stability. Okay, do you know what is shelf life and why we use this term shelf life? See, shelf life is something, the product which we used up to what time limit, and up to what time limit it remain contaminant free. Okay, the term very important, contaminant free. So this shelf life is basically up to what time period this particular product remain contaminant free, and up to what time this remain contaminant free, only only up to that period we can use that particular medicine or any kind of product. So these are the basic major areas of quality control, like good quality and nature, physically and chemically pure, contain amount of ingredient as stated in the label, it to be effective after administration, and it retain quality in terms of shelf life or stability. Now, what is the importance of quality control? What do you think? Like we studied about what is quality control, what are its major areas, what is quality assurance, and etc. So now we are going to know what is actually the importance of this. So the very first, if we talk about it, essential operation of pharmaceutical industry in order to deliver a quality, safety, and zero defect product to an economic rate. This is very important. Like whatever we manufactured, we need to like what we are manufacturing for what? For the patient, for the consumer. So we need to maintain that whatever we are delivering to the market, it should be defect free. Okay, so that the peoples can start using it, and it's also a source of marketing as well. So this is a very essential operation. Second, in order to maintain both the parameters of qualitative identification and quantitative determination. Next, the procedure and standard are prescribed in the pharmacopoeias published by the respective government of almost all countries. Like if I am manufacturing something, our government describe the board, that this board is giving license to this industry, this to this industry, this to this industry, etc. So for if we are starting any pharmaceutical company, we need to go with the board which can give us a license to manufacture that particular medicine and then market it for that. So we need to mention the procedure of its manufacturing very clearly, and its standard, like at what standard we made this medicine. Then they check it that is it as the reliable as consumer patients are required or not, and then they give us the license. Quality control is an essential drug must be marketed as a safe and therapeutically active formulation whose performance is consistent and predictable. Exactly, this is consistent means we need to change it as per the requirement. Like we cannot go with the same procedure all the time. We need to be consistent and predictable. Like we need to go with what are the pros and cons this medicine is have, what updates we require, or what updates or what remanufacturing is required in this. We need to go with that. New and better medicinal agent are being produced at an accelerated rate. The very important thing, if we produce our product or medicinal agent at an accelerated rate, then only we give it an affordable rate in the market. At the same time, more exacting and sophisticated analytical methods are being developed for their evaluation in order to minimize or eradicate the mischiefs or dangerous contamination pharmaceutical produce. These principles are very much essential. Like we need to ensure that whatever we are manufacturing, it's remain contaminate free. Because if any time contamination took place in that particular manufacturing, it's going dangerous for the patient or for the consumer. So we need to maintain maintain the environment where we are manufacturing completely contaminant free. And even time to time we need to ensure that the place where we are manufacturing and the product which we are delivering to the market should be contaminant free, so that the patient and consumer can effectively use it. Now, what are the methods used for quality control? Like what? So basically, there are two methods which we use for the quality control. The very first is qualitative analysis, and second is quantitative analysis. The very first we talk about is qualitative analysis, and second is quantitative analysis. Now we talk about qualitative analysis. What is qualitative analysis? This qualitative analysis is also called physiochemical method. Okay, physio include physical and chemical is completely indicate a chemical. So it's a combination of physical plus chemical method. So now very first, it include determination of a specific gravity. So what we are going, the very first thing is specific gravity. So what is specific gravity? It is the ratio of density of a substance to the density of a given material. Like whatever substance we are using or the material we are using, we need to find its specific gravity, and there is a formula to find that density of substance upon density of that material, whatever we are required. So by this formula we calculate the specific gravity. Second, density. Density we already know, mass per unit volume, mass upon volume. We can calculate density from here. Viscosity. Viscosity is resistance to the flow of liquid. Like whatever material or the substance we choose, how fluidity is present in that particular material is called its viscosity. Again, next, surface tension. Surface tension, cohesive force between liquid molecule. Now what is cohesive force? See, cohesive force is a kind of force which can hold the liquid molecules together. Like if we talk about water, in water, the force which is present between one water molecule and another water molecule is called cohesive force. That's why water, if we flow water in north to south direction, then the entire water flow will become from the north to south direction, because the water molecules is having a cohesive force. So we need to go with that force as well. Refractive index is the ratio of viscosity, ratio of velocity of light in a vacuum to that in a medium. Like if we find the refractive index of that particular, so we need to go with the ratio of velocity of light. Refractive index is what? Velocity of light in a vacuum upon in a medium. What is whatever with the velocity in vacuum upon what the medium. Medium might be air, glass, and water. Whatever with the medium, we need to go with that. Like if we talk about the refractive index of air, so it is 1. If we talk about the refractive index of glass, that is 3/4. If we talk about the refractive index of water, it is 4/3. So these are the some refractive index which we know. And if we talk about the refractive index of a vacuum, that is 0. So we need to know what is the refractive index of that particular substance or the material which we are using. And then last is optical rotation. That's the test of purity of a material, ability of a substance to rotate the plane of polarization. So what is this plane of polarization? Plane of polarization is something which is having the tendency to rotate the beam of light completely. Like if it is passing from one medium to another, if that light passing from here and here, we are having a plane of polarization. and then it passes through the next medium. So the light, when it passes from this medium, it can completely rotate the beam of light, and when transfer it having an opposite thing. Like, taking a simple example to explain your plane of polarization, when we saw the image of our hand in the mirror, in a simple plane mirror, we always size it opposite to our left hand, and if we are saying the right hand, it is completely opposite to the right hand. So that is plane of polarization. And test the purity of material by ability of a substance to rotate the plane of polarization of a beam of light that is passed through it, that particular beam of light, and all types of volumetric analysis. So these are the determination, like before starting the procedure, what thing we need to be go with: a specific gravity, density, viscosity, surface tension, refractive index, and optical rotation. Now there are some other instruments as well, like potentiometer, conductometer, polygraphy. These. Now we talk about the potentiometer. It can indicate electrode respond to a change in the activity or effective concentration of the analyte. Electrode respond, like we know we are having two electrodes. One is cathode and second is anode. So how these electrodes respond in a particular solution, that is called potentiometer. Okay. And then second is we go with the conductometer. Conductometer determine the total conductance of a solution. See, what is conductance? If we talk about the conductance is something which we called, we talk about the conductor. What is the function of conductor? If we talk about conductor, so conductor is basically which can conduct electricity. And why it conduct electricity? Because it is having free electrons. So just because of these free electrons, it is having the power to conduct the electricity, and that is called total conductance of a solution. And then polarography. Polarography is an electrochemical method of analyzing solution of reducible or oxidizable substance. Like, it is a method in which we can analyze any solution, like how that solution is reductible or oxidizable. We already know reduction and oxidization. So that how reductible or oxidizable that particular solution is, we can test with the polarography. Then we go with the colorimeter. Colorimeter determine the quantitative estimation of water. Quantitative estimation of color, sorry, it's color. To know the color of that particular substance, we go with the colorimeter. See, here you can see this is the potentiometer. The very first is potentiometer, second is conductometer, third is colorimeter, and fourth is polarography. These are the methods which we studied right now. Now, a spectrophotometer. Again, what is a spectrophotometer? See, a spectrophotometer is an instrument which we use to measure the intensity of light, basically. Okay, on the basis of wavelength. The principle of a spectrophotometer is based on the wavelength, basically. And next is fluorimeter. It can identify the presence and amount of a specific molecule in a medium. Like I as already told you about the medium, we are having medium like air, glass, and water. These etc. are the mediums. So in these vacuum is also act as a medium. So what medium that these mediums having some molecules or not, for detecting that we use fluorimeter. And flame photometer, it is an inorganic chemical analysis to determine the concentration of certain metal ions. Again, not all the metal ions, it is clearly mentioned certain metal ions, that it can detect only few of the metals ion. In that particular medium, either air, glass, water, vacuum, does they have some metal ions or not? For detecting this, we use flame photometer. See, here you can see this. The very first is spectrophotometer, second is fluorometer, and third is flame photometer. Now, this was a complete. Here we can completely go with quantitative. See, We first of all we look at the methods used for the quality control. I already told you we are having two methods: qualitative analysis and quantitative analysis. Qualitative analysis is also called physiochemical method, like physical and chemical method. So we saw the points. Basically we look at a substance or particular material: specific gravity, density, viscosity, surface tension, refractive index, optical rotation. After that, how we can detect these whole thing, we use few instruments like potentiometer, conductometer, polarography, colorimeter, spectrophotometer, fluorimeter, and flame photometer. So this is all about our qualitative analysis. Now we go with the quantitative analysis. Now what is this quantitative analysis? Quantitative analysis basically means when we can separate the molecules on the basis of their size, shape, gravity, etc. in these methods. So it is a separation technique. Quantitative analysis is what? It is a separation technique. Like chromatography, as we already know, chromatography, the function of chromatography is what? To separate the molecules on the basis of their size. It may be HPLC, HPTLC, TLC, and determination. These are the name of chromatographic, like TLC, thin layer chromatography. So these are what? These are chromatographic types. Determination by weight, like gravitometric precipitation method. Apart from the routine qualitative and quantitative analysis, pharmaceutical product are also evaluated for their quality. Obviously, whatever with the pharmaceutical product we are manufacturing, it should be evaluated on the basis of their quality, like how we can manufacture it and on what standard, what criteria, what protocols we follow to maintain its standard. We go with that all. So there are tests like disintegration, dissolution, hardness, friability, weight variation, content uniformity for unit solid dosage forms like tablet and capsule. If we manufacture any tablet or any capsule, so we need to test all these. To test the disintegration of tablet, test the dissolution of tablet, test the hardness, friability, weight variation, etc. of that particular tablet and capsule, and maintain it as per their instructions. Bioavailability studies, pharmacokinetic studies, biological assay, which we call bioassays basically, and microbiological studies are also carried out. Microbiology studies carried out just to know the contamination. Okay, that the tablet or a capsule which we manufactured, is it contaminant free or contaminated? So for that we need to go with the microbiological studies. Here you can see the instruments which we used in quantitative analysis. Now, as I already recently I told you about disintegration, dissolution. So now we are going to know what disintegration is and how we perform this disintegration, dissolution, etc. So disintegration testing measures the ability of a tablet to break down into smaller particles or granule. Break down into smaller particles. Again, a smaller particle. Like whatever we need to test, first of all we need to break it down into the smaller particle, or these smaller particles are also called granules. With the prescribed time movement of the axis from the vertical, when placed in a liquid medium at the experimental condition to allow the active drug to absorb in the body. Obviously, when we take something, we like if I'm taking a bite of food, so we grind it with the help of our teeth. So basically what we are going to do, we are just breaking the large molecules into the smaller molecules. The same with that disintegration process. What it should do, it can break down that particular tablet into a smaller particles or granules, so that they can easily absorb in our body. If we take it entirely, then it is not going to absorb in the body. That's why we need to break it down into the smaller particles or granules. Now second is dissolution. So what does dissolution means? Dissolution basically means measure the extent and rate of solution formation from a dosage form such as tablet, capsule, ornament, etc. Dissolution what is important for its bioavailability and therapeutic effectiveness. Like how effective it is. Basically dissolution means the molecule which we break down, how it easily solubilized in the media. That is called dissolution. It is a very simple term. Like in disintegration we break, like suppose this is a large molecule, and in disintegration we break it down into the smaller molecules. Now in dissolution, what is this? This is our media, and here it is the media. how easily these molecules are solubilized in this media. That is called dissolution. It's a very simple procedure. Okay. Now second is hardness. To determine the breaking point and structural integrity of a tablet and find out changes under condition of storage, transportation, packaging and handling before usage. We prepared that medicine. This is a medicine which we prepared basically. Don't go with the design. It's just a rough. So this is the medicine which we prepared. Under what condition we store it, how we can transport it, how we can package it, how we can handle it before usage. Like for adults it's a complete, for child they can take the half of this complete medicine. So this is all about how structurally we maintain this medicine basically. This is called hardness. Next is friability. It can test the durability basically. Durability is what? Durability is basically what, how long we preserve that particular medicine. It's called durability. Okay, like we that in medicine it is clearly mentioned you the expiry date. You can use it till this time period only. So that is its durability. That way it can remain, that particular medicine remain contaminant free up to that time only, and after that it is not for the usage purpose. We need to discard that medicine. So that is its friability. That clearly mentioned test the durability. Durability is the ability to withstand wear, pressure or damage of a tablet during transit, and the testing involves repeatedly dropping of a sample of tablet over a fixed time over using a rotating wheel with a baffle basically. Like at what temperature, under what pressure and under what conditions we preserve that particular medicine is called friability. Now weight variation, if we talk about the weight variation, it is a test would be satisfactory method for determining drug content uniformity or drug distribution. We already said that medicines are drug. So in particular medicine, in an individual medicine, what amount or what content of particular drug is present, that is their weight variation. Like if we are making 200 mg of medicine, so for making this 200 mg of medicine, how much drug content we are required? Like if I said 20 mg, or I'm saying like 10 mg if we are making for children, 20 mg if we are making for the adults. So this is their weight variation, how we distribute the drug and on what how we distribute the drug in particular medicine. That is called weight variation. Weight variation test is applicable when the tablet containing 50 mg or more drug substance. Okay, it is clearly mentioned we can weigh this test only if the medicine is having 50 mg of drug content. Like below this we are not going to perform this test. Drug substance, or when the drug substance represents 50% or more dosage form unit. Again, I'm saying here like 20 mg or 10 mg, they clearly mention the drug limit should be 50 mg or more than 50 mg. Then only we can perform this weight variation test. See, content uniformity. Content uniformity is again a test used to evaluate the equality of a dosage of a pharmaceutical drug. During the test, 10 tablets can be taken randomly from a sample in order to determine the amount of active pharmaceutical ingredient in them. Like this is just to know the variation, that the medicine which we have manufactured, is it having the same result or not, or what we select it randomly. Like we are not going to select it that with these 10 pieces we are taking or these 20 pieces, we can select it at random. Here is an example. You can see we can select the 12 sample basically, and the label amount of drug is 15 mg in each. Here it is clearly visible that in each medicine we just mention the batch code on each medicine, F1, F2, F3, F4, etc. But the drug content in each of them is same, that is 50 mg. Active drug content, see there you can see the variation in each medicine. 15.76, 14.9, 14.9, 14.92, 15.56. So here you can see the variation. Like actual drug content is not exactly same as the labeled amount of drug. So here you can see we mention on the medicine 15 mg of drug, but here the variation is basically vary. Someone is having 14.95, someone is having 15.01. So yeah, actual drug content vary. And here this is the result. This is content uniformity basically, just to check how uniform our content is. Now errors in pharmaceutical analysis. Just give me some time, we will continue it. Okay, this is our last topic, errors in pharmaceutical analysis, like how we manufactured and etc. We saw there how we qualitatively, quantitatively analyze it and all. Now what errors now pharmaceutical analysis is having? Like the very first is ultimate, in pharmaceutical analysis, ultimate result of analysis is important for accuracy and reliability point. Obviously if we are making something, manufacturing something for the accuracy and reliability purpose, so we need to make sure that the consumer sorry, the consumer which is consuming that particular medicine, it is completely accurate and reliable for that. So that is very first thing. The term accuracy refers to the agreement of experimental result with the true value, and it is usually expressed in terms of error. Precision is defined as a degree of agreement between various results of the same quantity, that is reproducibility of a result. And these basically are the two sources of error, determined error and indeterminate error. These are the two errors we are having. Like some errors we can determinate, and some errors we cannot determinate. This is all about determinate. Like obviously every error should not be determinate. So the error which cannot be determined is called indeterminate error. So these are the two sources. Hopefully you can understand the basics of pharmaceutical quality control. This is all about how we can control, what are the sources of error, how errors can be determined in pharmaceutical industry. Hopefully, thank you so very much.